Well, it's my great pleasure to introduce our research program here in China. We name it the Atlas of Blood Cell. So given the time limit, I'm going to briefly introduce you how the ABC initiative was established and a quick update on our ABC research in China. Finally, I'm going to use the ABC database to answer our biological question in the context of bone marrow transplantation. So I don't have to go through each detail of this technology aspect. You are all experts, much better than me. But in our research, we choose the single-cell RNA-seq as the most, the best defined the RNA sequencing technology. And I think we should give sufficient credit to Fu Tang in Beijing University. And most of people in China follow his protocol in the last several years. As you know, later on, Guo also established high throughput new technology in RNA sequencing technology. So we are expecting more even broader application of his new technology in the field. So why we want to focus on blood, and not because I'm the hematologist. I think simply I think blood is easily to obtain. It's a circulating cell type. Blood can sense our wellness and disease in our body. So after following the human cell atlas, so giving our limited resource and more focused question, and we established the molecular mapping program in the blood system in 2017. So this project was initially supported through Chinese Academy of Medical Science and following by multiple national major key research projects. I think one area I have to emphasize in the developmental hematopoiesis field led by Professor Bing Liu, I think really took full advantage of this technology, have moved this field into really front line. And of course, we have very active international collaboration with many countries globally. As I told you, we have established the comprehensive the RNA mapping in the mouse system. Now we're also moving on to other aspects, including some epigenetic landscape. So probably I'm not going to much more detail, rather than give you some example, highlight some the story being accomplished in China. So I think Bing Liu actually led us to trace the hematopoietic stem cell formation back to a few years ago, and I think that based on the seemingly limited data sets, I would say high quality single RNA-seq data, they follow up with additional two stories. One is to show embryonic endothelial evolution towards the first hematopoietic stem cell by the same data set. Again, and Bing and Yulan and Jia, they also combine the single cell profiling, particularly to look at the long-coding RNA, and to also was able to the map, the hematopoietic hierarchy. And in addition to that, they also showed Age19 is pivotal for the embryonic stem cell development. And I think that in our institute, that one group focused on neutrophil led by Humboldt Law. Actually, they recently published a paper in Nature Immunology showing the heterogeneity in a neutrophil compartment and under the both homeostatic as well as infection condition. So let me switch to the human data set. And recently, we also just complete the study by looking at the 32 immunophenotype purified human blood cell population, and then use the again SmartSeq2 to map those population. And our attention was to use transcriptome as a new definition to define different subset of human blood cell population. Here is a website you can look at into, and again, you are welcome to use the data, and hopefully we can optimize the data together to make the better tool for our community. And here's a summary of our ongoing ABC study mostly in our institute, from the normal blood cell type to the blood cell type in variety of human disease. I think you will see them a lot of story coming out in the next few years. And one example again is from the Bing Liu's group in Beijing. They traced the first human hematopoietic stem cell in human embryo. I think it is a landmark study and published in Cell Research last year. And shortly after that study, actually, they also actually showing the lymphoid region in the human embryo. And again, Bing Liu, in collaboration with Florent and in Singapore, that they define a human macrophage and the region in the human embryo. And the one key point is, again, they define the resident macrophage in a variety of human embryonic organ rather than from hematopoietic stem cell region. And they also revealed the cellular heterogeneity and normal subpopulation within the immune function during human megakaryopoiesis. This study, and led by Jiaxi Zhou in our institute, was just accepted in Cell Stem Cell actually yesterday. And there is another coming paper, and also they just submitted. And back to the disease, actually, we also noticed a study from the Zaijian Chen's group in Shanghai. They used the SmartSeq2 and in conjunction with 10x technology for the first time, they defined the new cell population in the leukemia cell type called M2 cell subtype, and molecularly, marked by and the chromosome 821 translocation. And also they link the data with the clinical outcome. I think that was a really interesting study you should look into. And there's a coming paper in the cell discovery led by Jing Wenfei in Shenzhen. They bravely, actually, they handled the largest set of data set in normal human bone marrow as well as 7 leukemia patients. And from the study, they show, trying interestingly, they were able to map each individual leukemia patient into the normal hematopoiesis. They were able to tell how each individual leukemia may likely originated from the correspondent stage in normal hematopoiesis. And I also show you some example. We apply our normal data set into the COVID patient. And the first study we did is to collect the blood cell from the 12 patient, actually 12 patient at a different stage of the COVID-19 in comparison with 3 normal individuals. Here's a summarized result, and we were able to show the nice correlation with clinical routine data, such as blood count. In the meantime, we also was able to define a new change of the subset of the immune subtype in the disease. Lastly, more importantly, we were able to use a single cell iron signature to correlate with the disease severity. In other words, we were able to predict who is going to likely progress into the critical type of the COVID-19. That's the purpose of the study. So another unpublished data is to collaborate with Zheng Zhang in Shenzhen. We were able to really define for the first time to answer the question, why the lymphopenia is often seen in the COVID patient. Also to explain why and the myeloid transfusion program often activated in some of the severe patients. So this study is still unpublished. I just briefly show you the result. So in short, a fine single-cell transcriptomic atlas of human blood system has been established in addition to the mouse map. The website is available to our scientific community. So our human ABC data have been applied to the variety of the medical condition in condition to the normal developmental hematopoiesis. I think this suggests is a greater potential in future translational medicine. Do you have time? Yeah, I have some time, I guess. Well, lastly, I want to use a few minutes to show a single story based on our mouse ABC data to try to answer the question and how the stem cell behave right after transplantation. So as you know, that nobody knows what happened to the stem cell that are just transplanted into the irradiated recipient shortly after transplantation, such as within the first week. So about several important questions you may ask. So what's the cell fate change? How the differentiation trajectory look like? And finally, how the stem cell pool is finally established. So with this question, I think it's difficult to use our classical functional study, such as transplantation to address the question, because transplantation assay is a retrospectively assay. So also it's difficult to use immunophenotype to define the cell type. As you know, some of the surface marker, such as c-KIT, will be downregulated immediately after transplantation. So we therefore employ the transcriptome-based definition to address the question in our field. As the previous speaker already nicely highlight and also emphasize the paradigm shift in hematopoiesis field. So we want to use the transcriptome definition to trace the regeneration of transplanted stem cell in the recipient. The first week, second week, third week, also the time after the first month. So here's one experiment that we did for the first time. So we purified the highly purified stem cell and then transplant it into the total body irradiated recipient. We harvest the GFP marked stem cell at day 1, 3, 5, and 7, and to see how the stem cell phenotype may likely change or expanded. As you can see from the summarized graph on the right bottom, and before we transplant, we see almost a homogeneous stem cell before transplantation. After transplantation, we see the drop and the dramatic drop of stem cell phenotype in the recipient. Instead, we see the MPP, the multipotent progenitor, become the dominant population. And even more interestingly, we even see some of the committed progenitor cells, such as the progenitor through the erythroid cell, also myeloid progenitor, being appear even at the day 1 after transplantation. So the real biological implication to be further defined. So we continue to trace the stem cell differentiation, also proliferation after the first week. But in this case, we use a much better, assay system. We use a single cell transplant as a way to answer our question. So with this very, very clean, fine system, we're able to absolutely quantify how many stem cell progeny being generated from each of the stem cell. Also, what kind of cell being generated. So with this experiment, as you can see, from the one single HSC, you can see variety of different type of progeny being generated in different recipients. And after 3 weeks, and I have to emphasize, within this time point, from the 1 week to 2 weeks, 3 weeks, we virtually didn't see any we didn't see dramatic expansion of HSC as marked as red color. This is quite surprising to us. And instead, we see the more differentiated cell appeared in the marrow. So after 1 month, we see the dramatic increase of the stem cell as measured by flow cytometry, also measured by functional definition. And when you purify the HSC, use the same phenotype before we transplant, we see within the immunophenotype defined stem cell compartment, we see the variety of transcriptome defined cell type and in so-called stem cell compartment. That indicate the true stem cell has been compromised. Instead, more differentiated, even more committed precursor already appeared in the immunophenotype defined stem cell compartment. So
2 minutes, Professor Tao.
Okay.
Yeah, thank you. Just one more minute, if that's okay. Thank you.
Okay. So one conclusion from our studies, we see the better agreement between transcriptional definition and the functional assessment. Okay. Okay. So I think that for the first time, we unreveal the black box of transplant stem cell in the irradiated recipient. So I think we call this functional immunophenotype transcriptional definition as the new approach to study stem cell. So this study by the number of people in my group, also wonderful collaborating bioinformatics field, as well as a number of clinicians in different hospitals. So you are welcome to attend our International Forum Stem Cell in next month. So I think stay on. So more information coming out. Thank you very much. Thank you.
Thank you, Professor Tao. Are there any questions for, well, while we're waiting, if there's any questions for Professor Tao, I have one question. Yeah.
Go ahead.
So thank you. That's a fascinating work. And it reminded me of some of the early work from Ihor Lemishka and other people with the retroviral barcoding of stem cells and transplantation in mice. They found that there was just one clone, this fit clone, that was contributing to most of the lineages in the mouse. And there was this other beautiful study by Gerald Dehaan with barcoding. It was in, I think, JXMed or Blood, and it's not as famous a paper. But when they transplanted long-term stem cells, only a few were contributing to peripheral lineages. But when they went back to the bone marrow and grew up colonies, they found all these other barcodes. So that when they transplanted it, they got into the bone marrow and then they just sat there and they didn't do anything. And when they hit with GCSF, then suddenly they started producing lineages. So it's interesting, this idea of fitness upon transplantation in that first round, what's active and what becomes quiescent, and then maybe what signals in the future can revive them if they're silent and sitting in the bone marrow. Sorry, that was a general comment, but I'm wondering if you have any thoughts on what proportion of stem cells you transplant are present but silent versus present and active.
Yeah, this is our, the great question. Of course, we always trying to see that after transplantation, what kind of stem cell remain quiet and what stem cell activity. In this case, if you remember what I show you, all the single cell transplant study, I think all the cell will go to cell cycle, right? Otherwise, we would not able to see any progeny. So in terms of the differentiation versus proliferation, one most striking even surprising data to me is that the stem cell tend to differentiate first rather than self-renew and as a major event. So this is, to me, I think is an unexpected result. In my mind, and the stem cell undergo dramatic expansion, self-renew, and then differentiating. So I think our study strongly suggests differentiating first, and then how the stem cell accumulated, that's a different question. From our current data set, we still cannot tell you how the stem cell pool eventually, after 1 month, accumulated.
Yeah.
Okay.
Yeah, so that'll be interesting to dissect all of that. Yeah, fascinating work, Professor Tao.